Hello, my name is Elena Olson. Today I will be talking about effect of cetylpyridinium chloride on the microbiota of Salmonella typhimurium and Salmonella infantis inoculated chicken skin. Firmly attached bacteria are the bacteria that cannot be eliminated by rinsing methods. Many studies show that bacterial cells that are attached to poultry skin are unreachable by many various antimicrobials. More so, chicken skin displays a greater buffering effect than other poultry parts. So the durability of salmonella on fresh manufactured poultry skin and the difficulties of eliminating that foodborne pathogen may be due to the complexity of eliminating these attached cells by commonly used washing and rinsing strategies. Post-chill antimicrobial application has demonstrated the ability to produce the most reduction of these foodborne pathogens, and over 10 antimicrobials are approved in the poultry industry during this step, with PAA, peracetic acid, and CPC, cetylpyridinium chloride, as their two leading agents. PAA is an organic acid and is considered an ultimate antimicrobial agent in the industry due to its high oxidizing ability, and previously shown a great success at reducing pathogen numbers on chicken skin. CPC, on the other hand, is a quaternary ammonium sanitizer or surfactant that showed ability to disrupt the bacterial respiratory cycle, as well as showed ability to change the morphology of salmonella and reduce its numbers on chicken skin. Salmonella infantis is becoming a significant problem in public health as well as food safety. Thus, the current study concentrated the potential beneficial antimicrobial effects of CPC on Salmonella typhimurium and Salmonella infantis when inoculated on chicken skin compared to PAA. As in part one of our study presented in the same conference by Dr. Dana Dido, indicated that CPC treatment may potentially reduce the invasion of Salmonella infantis, the part two of this study concentrated on determining the response of the microbiota of the chicken skin inoculated with Salmonella typhimurium and Salmonella infantis to CPC and PAA when those were applied as short duration antimicrobial dips. Typically, PAA and other products are tested against Salmonella typhimurium in the industry. Thus, we use the reservoir as a control measure in the study. And our hypothesis was that PAA and CPC will have similar results or effects on the microbiota of the inoculated chicken skins. So as a study design, our methods were adapted from Lillard et al. 1985. And the following experiment was conducted over two trials with different inoculum used during each separate trial. A total of 10 whole fresh chickens were obtained from the commercial pilot processing plant, and the skins of the breast were removed aseptically and cut into 4 by 4 centimeter squares. 5 of those squares were then randomly assigned to 5 different treatments, which are described here in the lower right corner. A frozen stock of Salmonella typhimurium and Infantis were selected to be resistant to 64 micrograms per milliliter of nalidixic acid. And then the chicken skins were inoculated with appropriate reservoir at 4 degrees Celsius for 30 minutes in flasks and stirred on stir plates. Skins were then shaken using sterile forceps for 30 seconds to remove any excess fluid, and any remained bacteria were considered firmly attached. So there were a total of 5 treatments. All treatments were administered at room temperature for 20 seconds. Skins again were then shaken with sterile forceps for 30 seconds to remove any excess fluid, and the remained bacteria were considered firmly attached. Following the 20-second treatments, the skins were then moved into buffered peptone water and stomached. The resulting rinsates were then saved for the downstream analysis. The DNA was extracted for all the collected rinsates from both trials using Fast Minis Toolkit by QIAGEN. 16 sRNA gene sequencing libraries targeting V4 region were then created. These V4 regions of 16S rRNA gene were then amplified and sequenced, and similar sequences were grouped into OTUs, or observed taxonomical units. And then using a SilvaGenes database, we were able to identify these observed taxonomical units and more so visualize them, visualize the output that was generated via taxa plots, And further, we went to statistically differentiate the taxa using analysis of composition of microbiomes, or ANCOM. This brings us into the results. Biodiversity metrics include alpha and beta diversity. Alpha diversity looks at the difference within the samples, whereas beta diversity considers both, considers the differences between the given samples. And so evenness is a biodiversity metric of alpha diversity, and evenness describes relative abundance of different species within the sample, or how close in numbers each species is within the sample. So here, the pilose evenness for the treatment groups was different within the skins treated with PAA and no treatment control, as well as PAA was different from the tap water. And when we looked at the inocular groups, the species evenness was different within the skins inoculated with Salmonella infantis and Salmonella typhimurium, as well as there was a significant interaction between treatment and inoculum. Shannon's diversity, or richness, describes richness of the sample, and it is as well an alpha diversity metric. Richness describes a number of different species present within the sample, and Shannon's diversity index describes how diverse species are within the community. And here, similar results were observed as were seen with the pilose evenness for the treatment groups. The richness was different within skins treated, again, with PAA and no treatment control, as well as PAA and tap water. For inocular groups, the richness was different within the skins as well, within the serovars, and significant interaction between treatment and inocular was observed. The beta diversity metrics were assessed with quantitative indicators such as Bray-Curtis and weighted unifrac distance matrix. Using permutational multivariate analysis of variance or PERMANOVA, we were able to test the null hypothesis of no difference in dispersion between the two groups. So Bray-Curtis is a compositional dissimilarity matrix that measures dissimilarity between the two different samples based on abundance. In this study, the microbial abundances between CPC and PAA were not significantly different, as you can see the overlapping of the circles here, but they were all different from the groups, from the control groups. As well as there was a significant difference between the microbial abundances associated with inoculum. Weighted unifrac is another compositional dissimilarity matrix that compares the similarity between given samples based on abundance, just like Bray-Curtis, but also incorporates phylogenetic distances. And here we can see that in the treatment group, no inoculum, no treatment control was significantly different from the rest of the treatments, inoculated treatments. And here, and as well as no treatment control was different from PAA. And phylogenetic diversity between Salmonella serovars was different as well. So while beta diversity tells us what is different, or that is something is different, ANCOM, or analysis of composition of microbiome, tells us who is different. So here we are looking at the differences, taxonomic difference, relative abundance differences within the inocular treatments. And the interesting observation that was observed that Campylobacteriota and cyanobacteria were significantly associated on the phyla level, and as well as Bifidobacterium and Leuconostoc genera were associated with both serovars. Bifidobacterium is used as a probiotic in the industry and actually been seen to be antagonistic to Salmonella. And here we can see that it is associated significantly with both serovars and more so with Salmonella infantis. Leuconostoc is a prebiotic that promotes Bifidobacterium growth and doesn't promote Salmonella growth. Again, a very interesting finding. And here we're looking at the ANCOM results for the treatment groups. And we can see we found over 30 significantly different phyla associated with all the treatments. All of those phylas are commonly associated with the poultry gastrointestinal tract. such as proteobacteria, firmicutes, bacteroida, and campylobacterota. And more so, we saw that bacteroidota relative abundance was a little bit higher with CPC treatment than PAA. And on the general level, we saw Brochathrix species, genera that's associated with the spoilage of meat stored at the cold temperatures, as well as bad odor of meat. And Chivinilla genus includes over 70 species, which some of them are indicative of food spoilage and caused microbially induced corrosion on steel surfaces. And the last one is Yersinia is a gram-negative bacteria from the phylum of Proteobacteria that's been previously isolated from broiler chickens and has been associated with high resistance to antibiotics. So at the end, Salmonella tolerance to acid differs between strains and serovars, and previous research showed that Salmonella infantis to be more acid tolerant than other several Salmonella serovars, including Salmonella Typhimurium. The use of organic acid in the poultry industry may contribute to selection and development of acid tolerant strains. So surfactants such as CPC, they increase the solubility of chemicals and their penetration. So the future study should concentrate on combinatorial effects of CPC and PAA to potentially reduce skin buffering effect, as well as avert the emergence of acid-tolerant bacteria. And in conclusion, CPC used as a short duration dip is an effective antimicrobial alternative to PAA, and as well as it's capable of producing a stable microbiota on poultry skin. So I would like to acknowledge Safe Foods, Animal and Dairy Sciences, and Meat Sciences of Biologic Discovery at UW-Madison. Thank you.